Hi, this is for Nuclear Medicine 3011A. I'm Dr. Stein, and this basically is a synopsis of week one. It will probably take anywhere between 1 to 3 video presentations. There'll be some questions to look at to review throughout, but I will include those at the end as a supplement. In other words, I will add them just answer for your own benefit uh, and look up more than anything else. And I'll incorporate those uh, in a separate document that is included with this particular uh, online course. So, my portion of this particular course handles the biology portion of it, where the other recordings are made by Dr. Gavin Buxton, who is a physics professor. I'm a biology senior lecturer, and we both use our areas of expertise. My background is medical and clinical, and his is a wide variety of interesting topics in physics. So I will begin now, and this is the PowerPoint, and I will always, and it should already be on the Blackboard shell, is the PowerPoint that we'll be referring to start with. This comes from, as you can see, from the Maribin. I believe it's pronounced home. Everybody pronounces these names differently, by the way. And this is from their seventh edition, which is the textbook that was used originally. There are newer versions of this that are available, but I prefer to use this. And additionally, for some of the other areas, I've downloaded some other PowerPoint materials. Uh, there are a lot of good PowerPoint resources online, a lot of slide sources that you can find now that you can use, and if you can use more extensively, they typically come with some degree of advertising in them during the slides, but that's not a particularly big deal. That there, so you'll see that we will use these incorporated, and they're very useful, particularly in this area of uh, radiation biology, where there are not necessarily a ton. There's not really a lot of textbook resources other than ones we are using, but that's actually very good, and you will see that, and it's easily acquirable. That radiation biology textbook that's been around for a fairly long time is still very, very relevant. Perhaps the numbers vis-a-vis physics and radiation exposure may have changed. I do not know. That's not my area of expertise. Uh, so perhaps with the degree of technology that you'll be employing in this particular course, because the technology, when we're using it, I refer to them as advanced imaging techniques. That's my frame of reference. So it can start with MRIs and CT scans, either ones that are enhanced, PET scans, and a variety of other types of nuclear imaging, bone scans, which I'm very, very familiar with, and the use of these radionuclide agents, all of which I know that this is somewhat redundant for you, but my portion is basically the biological, and we have to start somewhere, and we might as well start with the cell. So, fortunately, as I was saying before, that hasn't changed. We understand some things, but it really has not changed. So, I will begin forthwith. And I'll probably just conclude. I may just do this PowerPoint by PowerPoint. There's four in this sequence, and frankly, I'm not going to spend a lot of time with the third one. This is, and you probably have had this before. You may have actually used this or a very, very similar textbook for anatomy and physiology or whatever your base course that you need to take this with is, and basically what cells are. So living things are made out of cell. We already know that, and it's all about the cells. Our focus is going to be on what makes the cells, let's say, vulnerable for us. But we have to know some basics on them. Why certain cells are relatively resilient in the face of uh, radiation exposure, and why other cells are not. So, and this deals a lot with structure and function and complementary, and I'll comment on that. And really the aspect, if you want to break it down, and as I was thinking as I was developing this presentation, the reality is that ionizing radiation, whether it's X-rays or other forms of radiation that we employ, both therapeutic and diagnostic, uh, basically they create an environment, and particularly with what we call toxic derivatives of oxygen. that are able to disrupt DNA strands. And we'll do a mini review of this as well. But DNA strands are all about what effectively the expression of our cells and how they function. So, and I'm fond of saying you are what your DNA says you are. Well, that's all well and good. But the fact is, we're breaking and damaging existing DNA strands in cells that are actively replicating, cells that are actively undergoing mitosis, asexual reproduction, whose sole purpose is to make identical copies. Then we're going to have problems because those are typically the most active cells. They're the most susceptible to damage. And when they are damaged, they develop errors in DNA from exposure to ionizing radiation. Those damages to DNA, when they are expressed, when that DNA is part of a gene that is expressed, whether it's a regulatory gene or a gene that expresses itself in proteins, it's going to lead to problems. It's either going to make proteins not form, they may misform, it may not have any impact at all. But the reality is it's a numbers game, ultimately. And the greater the degree of exposure, once it reaches critical levels, is going to induce a number of mutations. What's a mutation? A simple definition: an error in DNA. Doesn't, and again, you're going to see it, and we'll look at this again, that errors in DNA can be silent. In other words, they really don't exist. We either correct them or they're not included because of mechanisms that are there within the cell to prevent them, or a surveillance system that's known as DNA polymerase 3. And DNA polymerase 3 is a very advanced enzyme that's able to identify DNA errors and correct them. Very, very much so, uh, in a way that effectively what a DNA error is when you have the typical genetic sequence of DNA, A, T, G, and C, adenine, thymine, guanine, and cytosine, mispaired. Adenine is always with thymine, guanine is always with cytosine. If there's an error, if there's an adenine with a guanine, a thymine with a guanine, those are going to be mismatches. And DNA polymerase 3 has the remarkable ability to identify where the error is on which side of the strand, because effectively these are two identical strands, one right side up and one upside down, and replace the one where the error is and correct it effectively, cut out the error and put in a new strand. It's a remarkable process, and it's way beyond the scope of what we're talking about here, but just to give you an idea. Okay. So we have to look at a lot of what they can impact. So effectively, if these mutations are expressed sufficiently, they're going to at the very least alter function, and sometimes cause significant problems. And the worst case scenario, they will cause replication to effectively spiral out of control. When replication on a cellular basis spirals out of control, it just makes continually more and more mutations, and inevitably they lead to either destruction or, even worse, cancerous changes. And most of the time, it's lining cells, and we do a whole unit subsequently on tissues. And the problem with tissues, particularly lining tissues, epidermal tissues, and the tissues that underlie them, called connective tissues, they are active and replicating for the most part, and they can easily become cancerous. Here we go from here. So back to it. All living things are made ourselves. We have to understand it. There's lots of different types and sizes. You can just see here some function individually. Rarely, those are macrophages, white blood cells. They're more defending us than problems. Here's the difference between an epithelial cell, a lining cell, and a connective tissue cell. Lining cells are attached to one another, where connective tissue cells are not. Red blood cells function as a unit, and they're unique because they're really not even a cell because they've lost their nucleus. They don't need it, they just have to carry oxygen. More advanced cells, like muscle cells or nerve cells, are very, very elaborate, very, very developed to the point where they effectively no longer undergo mitosis. That ship has sailed, so to speak. So unless there's been damage in the intrauterine environment while the baby's developing, that's the distinction that's there. And we'll talk about that with what we call differentiated and undifferentiated and dedifferentiated cells. That's important. Fat cells are a storage vehicle. Okay, problems can occur in them because they're relatively simplistic. Again, white blood cells, problems can occur there. Sperm cells and eggs, very much because they lack a lot of the protections. They're very vulnerable. And everybody who's ever had an x-ray taken in a hospital or a medical unit, they're going to put an apron on you. Please inform us if you're pregnant. You're trying to protect gonadal tissue, eggs. You're basically ovaries and testes in that perform. So these are the parts we're going to start to look at. Uh, we're going to look at the membrane a little bit. It's pretty straightforward. It's a phospholipid bilayer. It's a boundary. It's flexible. It's dynamic. It's got proteins embedded in it, which you're going to see shortly, that have a variety of functions. And basically, those proteins act as a gatekeeper. They allow some stuff in and some stuff out, sometimes automatically, sometimes with signals. Sometimes they just interact with the outside environment and they change in such a way that they impact the internal environment. So they are pivotal. We have things that stabilize the membranes. They're basically fats because the interior part of the membrane is fatty, and cholesterol, little islands of cholesterol and similar subunits, play a pivotal role in stability, also opening and closing membranes. The cytoplasm is just, and a plasma is just a generic term for it's like a, we call it a solid gel. Some of it's solid, some of it's gelatin, some of it's solution, all of the things that are inside the cell. But the stuff that's gelatin and solution is really the cytoplasm. The stuff that's solid are organelles or little organs. And the nucleus basically directs everything from traffic to what's made to what's not made and to when the cell replicates. You've all seen this before. It's not simple. It's complex. But our focus is going to be less about a lot of these organelles other than their impact, but more about perhaps what goes on here in the nucleus and very much so how basically where adversity comes from might be the best way. So that's why I don't want to do the laundry list. Well, the mitochondria is an energy plant, and the smooth and rough endoplasmic reticulum, or just a passageway for proteins that are going to be either spit out of the cell or incorporated in the membrane or used in the lysosome, terms you've already heard before. And then there's lots of different fluids and things like that play a role outside the cell. It's called extracellular or interstitial, that surrounds the cells or outside of them. Plasma is just another fluid. Cerebrospinal fluid's a fluid. Different things we make, saliva, mucus. There's even material, kind of like a gel, that can be firm or not so firm or filled with air, etc. When we get to the tissues, you'll see that, but it holds them together outside the cell. And we call that the extracellular matrix. That's an intricate and an important part of connective tissues. And so basically the gatekeeper role of the plasma membrane. By lipid bilayer proteins, we call it fluid mosaics, famous term because it can change shape. It's fluid, it can open and close. It's fluid, and the proteins can to some extent move around. And that's where the mosaic comes from, different proteins in different locations. Sugars protrude, they're called a glycocalyx, which means sugar shell. And what's interesting about them is there's a lot of identity. These tiny chains of sugars protrude are what makes your blood type B and mine A, or someone else's O, or someone else's AB. And it's really an important part of our immunological recognition that's there. And as you can see, that and we'll see a lot of cell junctions. And again, some cells are very tightly bound, lining cells. Some are loosely bound. Some aren't bound at all. And there's a lot of variations. And this is just basically giving you and you can look through this. These are very hard to see unless you expand them, and I'm not going to try to, what the constituents are. And you can see the natures of these little sort of hexagonal structures diagrammatically are sugars that protrude. Most of them are six carbon sugars we call hexoses. You probably heard the names glucose and fructose are pretty much the basic members. There's others. The little yellow is the cholesterol, and these are the embedded proteins. And some are little openings. Effectively, it's a pore. Okay. Some of them change shape to allow stuff in and out. Some have things attached on the outside or the inside, so something can attach it here and cause a change in here. And what we don't recognize, with rare exceptions like a white blood cell, There's a fiber network inside that these attach to, and there's a fiber network outside. So cells rarely function independently. Again, macrophages are a little bit different. They're part of your defensive system. They go to the problem. I'm not going to do those uh, animations as part of this. I mean, you can see them well enough. So you can see the constituents. And again, what makes that bilayer interesting, it's water soluble on the outside because outside of the cell is a fluid base. Inside the cytoplasm is a fluid base, but the boundary is a little tiny layer of fat. Comparatively thin, but a sufficient boundary, and things that are, if they're not compatible with fats, in other words, they're small, what we call nonpolar molecules, and you all should know what nonpolar and polar means. If they're nonpolar molecules and they're tiny, they can get through. Polar molecules like water often can get through. There typically are little pathways for charged particles. Sometimes they get through automatically, sometimes they don't, but they can typically get through. Big things like proteins can't get through. Okay. And so what happens, these phospholipids are bipolar, so they have water soluble on one side, fat soluble on the lipid side. The lipid sides point toward the middle, and they effectively create the barrier. And there's other stuff. And again, the cholesterol is there. So this is what points out and points in. And these are sort of like, it's like when you were a little kid and you and your friends lay down and put your feet together. It's like made your feet move together. I did with my older sister, that type of stuff. And maybe you did something silly, but like it's like heads and feet and feet. And so and they're very heavy. Proteins are big. Ones that are within the membrane are integral, and often the fatty soluble portions, the amino acids that are compatible with that, are in the middle, and the ones that are water soluble are on the outside. And very often they have second portions that stick out or stick in, called peripheral proteins. And there's a subtle difference between them. And they have a lot of functions which you're going to see. And I don't mean to go through it because they haven't listed here. So if they have openings, either they're all the time, like a water pore, or maybe for sodium or potassium, or they have one that changes shape when something comes contact with it. We refer to those as transport proteins. They span it. They have a little water-filled channel so those particles can get through, even though this is membrane, but this bridges it. And every so often, the question is whether we need to impart energy. Sometimes if we don't, okay, we rely on diffusion. If it requires your channel, we call it facilitated diffusion. We don't have to add energy. If we have to add energy, there's often different reasons. We're either actively pumping out something against the grain. Remember, things that move in and out of the cells are based on something called concentration gradient. Shape, size, compatibility plays a role. But is there more outside than inside? And if there is, they're going to go from outside to inside. If we're going against the grain, trying to pump something out, when there's one in here and a bunch out here, we've got to put some energy into it. And there's exceptions, but it's not relevant. And again, they show you a lot of these little things. This is classic. Best example would be if this is a big protein like insulin that has receptors on almost every cell that we have. And if Phil hits this, and when something attaches to a protein, even if it's another protein or it's just a sugar or a charged particle, it changes shape. And when it changes shape, that shape change will be, it's like you touch somebody knocked on the door and you walks over to answer it. I mean, that's from the outside imparting a change on the inside. You have to get up, you have to walk over the door, you have to open it. Maybe look outside the peephole, make sure it's somebody you want to let in. But this is effectively what's happening with insulin, except you'll find that insulin sets off a chain reaction inside by an interesting pathway. So that's a classic chain of chemical reactions. That's receptors, and mostly in the gut. Okay, they have little enzymes. And so if this is inside the cell up here and outside the cell down here, here you can have sugars that come in contact with it, and the enzymes will break down or change the little sugars till they're very simple sugars. Because, and this is why we talk about being lactose intolerant. Lactose is a double sugar. It's too big. Can't get in the cell. So we have lactase, an enzyme that breaks it down into something called galactose and something called glucose, which can easily enter the cell. Now, if it doesn't and the lactose is still out there, then the lactose pulls water along, and it upsets your stomach because things move too rapidly, etc. And we do see a lot of that in the gut. And then this is what I was talking about before, cell to cell recognition proteins. And the recognition proteins are interesting because they're part of that sugar shell. And this is other cells can recognize them and bond to them. Other cells can recognize them and leave them alone, or white blood cells can not recognize them and attack them. And it's the basis of our immune system. It's the basis of ABO compatibility, blood types that are compatible. So someone could give me, or I could give someone else a transfusion. It's so important, and it's very subtle. I mean, if this chain had a little extra bump on it, they wouldn't be compatible. That's there. And then we have attachment proteins. So whether it attaches directly to the protein with other fibrous proteins or by some intermediary, it's like something on the outside can pull and tug on something on the inside. And that happens as tissue layers develop. In the skin, as the cells move farther away from where they start on the outermost layer called the epidermis, effectively they're growing away from their base and they're being pulled by the other cells and they begin to change shape. And that shape change brings about some additional changes as they grow away from their blood supply. And as you probably know, on your skin, when the cells get far enough from your blood supply, they start to die and eventually our outer layers are dead cells. And there's a lot of movement that's associated with this as well as holding shape, etc. These fancy names called CAMs or cell adhesion molecules, they hold cells together. Lining cells are held together tightly. Connective tissue fairly tightly. Your heart muscle very tightly. It's got series of these, and there's different categories. Ones that are like riveted are called desmosomes. They're the strongest. They really hold things in place. Ones are called tight junctions. Read that as watertight. The more you have of those, the harder it is for a water molecule, if this were two cells, one here and one here, to get in between them, the more there are. And then there's another one called a gap junction, which is like a little protein passageway that little particles can go through. That's how the heart muscle tells the next heart muscle, hey, time to fire. I fired, so now you fired. And bone cells can signal to one another to grow or not to grow. There's a lot that goes on. So here's the glyco, and you can have glyco sugars attached to proteins, sugars attached to fat, and then this is that whole idea of identification, self versus non-self. When you're born, your body has about 6 months to get to know what your cells looks like as your immune system kind of ramps up to speed. Those first 6 months, your mom's immune system is covering you. Then after that, those antibodies, the chemicals that she gave you, effectively dissipate. They no longer function. And we start making our own. But it's an opportunity for your body to identify what belongs and what doesn't. And again, this is an interesting nugget. Cancers change those markers so rapidly that it mutates so fast that a white blood cell, which is made to attack that particular mutation, fails because it's mutated once again, which is very interesting. Here are the tight junctions, desmosomes, and gap junctions I was talking about. You can read through this as easily as I can, and it's very light. We don't have to get into the structure. Okay, and again, you can see the signaling aspect. These are proteins that make a tiny passageway that'll spit out charged particles, for instance, in some instances, or other chemicals. Okay, and this is the idea of selective permeability. We're getting near the end of this first portion of the presentation. Selective permeability is such that some things can get through and some can't. Living membranes, they all have that. That's part of their identity. Phospholipid bilayer, embedded proteins, fluid mosaic model, by definition, selectively or semi-permeable. Again, we use concentration gradient. If it can get through the membrane by itself, like oxygen and carbon dioxide always can because they're small and they're fat soluble, we rely on concentration gradient. More oxygen in your lungs, less in your bloodstream in the lungs, they move. Carbon dioxide abundant in your lungs and the blood, not so much in the lungs itself in the air spaces, it moves the other way. That's respiration 101. And if we do have to use energy because we're moving things out in bulk, spitting them out of the cell or bringing them into a cell, endo or exocytosis, or if we are resetting cells that move, nerve and muscle cells are constantly being reset. They're constantly changing electrically. And so we are constantly, every time we have a thought, speak, move a muscle, that cell has done a lot of business. And sodium and potassium have sort of switched places. We have to pump them back out to reset them chemically. And to do that, more than half your energy goes to something called sodium potassium pumps, or basically these are sodium potassium pumps that reset the internal milieu, the internal concentration of the cell, and the external, as you will see shortly. So if we're relying on the concentration, we don't have to worry about, as long as we can get through, or there's a passageway, if we can get through the membrane, simple. If it gets through a passageway, facilitated, we have something helping it, facilitated, enabling it, a protein. And and those are what we call solutes: sodium, potassium, glucose. Everything is dissolved in water. Now, water has a concentration too. It's called a solvent. It's the universal solvent for our bodies. So a lot of times it's where the water is. So if you're dehydrated, your cells are thirsting for water, you put extra water in. And eventually the cells get a balance. If there's too much water in, basically your kidneys will take care of it and you urinate it out. The problem with osmosis is if we don't correct a situation that must be corrected. If we don't, if we're dehydrated and we don't replenish, we're in trouble. If we overhydrate and do so rapidly that we don't have a chance to get rid of the excess water, we're in trouble. So a lot of that has to do with what's called the concentration of water. So whether sodium and potassium are moving in and out of the cell is one thing, but water kind of balances out. And you'll see some graphics about this. So it's all about the concentration gradient, and basically it makes it how, when things are packed tighter together, they bump into each other molecularly, and they tend to move away from one another. And they show you this. It's like put a blob of ink in a beaker, and next thing you know, it'll be blue. Okay, a drop of blood in a beaker of this size, maybe 500 milliliters, that'll be red right away. Won't take long. Okay, and different things, size, temperature makes it faster. Smaller is faster. Higher concentration is faster. Eventually, things always move, but when they sort of, you have a balance. If there's 50 particles inside and 50 particles outside, it doesn't stop. You'll have one go this way and one go the other way. That's what's called equilibrium. And again, differentially permeable. This is the simple diffusion, oxygen, carbon dioxide. When we talk about facilitated diffusion, it needs a helper. Now, sometimes the channel by itself, insulin is the helper. It doesn't open the channel directly. It sends off a chain reaction inside. One of the things that it opens glucose channels to allow glucose to enter the cell. And in water, it's almost like we're just looking at the amount of water related to the amount of particles that are there. And that's why burns are such a problem, because we use fluids and electrolytes, and burns get infected very easily as well. So they'll show you some examples of the classic one: oxygen, carbon dioxide, steroid hormones, testosterone and estrogen. They get through easy. And again, a little water can make it way depending on it. And they're going to show you that to you, but I'm not going to do it. Facilitated means there's some kind of carrier, a channel for it to get through. They're very specific. If it's a sodium channel, it's got to be sodium. If it's potassium, it's got to be potassium, glucose, etc. And one of the problems is down the road is when they're all filled up, then it creates some problems. But that's really, again, not something we explore in this course so much. And we have different kinds of channels. Ones that leak, they're always open. Ones that are gated, they need something to open them. There's different things that open them. And I'll talk about that in passing. And again, water channels are almost always there. Aquaporins, pores, whatever you want to call them, works for me. And this is an interesting nugget. It's what we call osmolarity. It's basically how many particles per unit of water. Typically, let's say in a liter of water, if we have 300 particles, that's an osmolarity of 300. Typically, tiny particles, we call them milliosmoles. That's about normal. So water's just like having too much salt or having too much water. If there's more outside or inside, and it just moves the same way. And they have an interesting graphic. So here, This is solutes. This is potassium or sodium, and this is dissolved in water. Well, if you have an opening that the solute, the sodium or potassium, can move from, if there's more here and not so many here, and you magically open this door, eventually it'll balance out. And one will go this way and one will go that way, and it'll stay this way. But suppose you have the same situation and that little opening is only big enough for water. Well, what happens here, now we flip the switch in this membrane, again, more this side, but look at theoretically, if you have a whole bunch of particles and this same amount of water here, very few particles here, proportionally you have more water. So even though solute is more here, it can't move to balance. So the water has to move to balance. Okay. It's like when you eat a bunch of French fries with salt on them, and oh boy, am I thirsty. You got to find a way to balance it out. So we open the switch and the water moves. That's osmosis. So it creates a pressure of its own. Hydrostatic pressure is blood pressure. It's the force of water being moved. Okay. So that's really a form of hydrostatic pressure. Okay. So, and osmosis will play a role in that because you're putting more pressure on the walls of the cell. Okay. Osmotic pressure is being pulled in the opposite direction. Proteins in your blood hold the water in the bloodstream because theoretically, with blood pressure, you could push it all out of the capillaries, but it doesn't. So, and the two are always in balance, except when they need to move. So you're going to find, like if we get to a capillary bed, there's changes. When we get into the kidney, there's changes. Okay. And so that's all of it. And this is what happens if you get dehydrated, water's going to start to leave your cells, and the cells are going to shrink and they can malfunction. The membranes can lyse or split. And by the same token, unlike a plant, we don't have a cell wall. So if you're overhydrated, that cell can burst. Okay. So it's a difference between us and plants. The fancy name we give to this is tonicity. And what we use in the hospital, and if you're administering an IV, which many of you may be doing, and injecting a certain amount of contrast material, radioisotopes, etc., that you typically, the media we use is isotonic, which means it has the same concentration of solutes to solvent. Now, our body has a myriad of things: salt, sugars, other chemicals, hormones, proteins. We typically use what's called normal saline. What makes it normal, it has the same proportion of basically particles, not molecules, particles. Salt is a molecule. Put it in water, it makes two particles, sodium and chloride. Sugar is a molecule. Put it in water, you have one sugar. It doesn't split up. So it's different. So we most commonly use normal saline. What makes it normal is about 0.09%, which means 0.09 grams, teeny tiny amount, pinch of salt in 100 milliliters of water, about 3 1/3 ounces. Now, if you make it like seawater, hypertonic. If you make it triple distilled water, hypotonic, no solutes in it. And it's going to either make them go big or go small. And so this is what they're explaining to you about that. That's the classic experiment where if you put it around a red cell, stay the same, isotonic. If it's too salty, this is called crenation. And it pulls the water out and you get little bumps under a microscope that when we see them with a light microscope, this is an electron microscope, they appear like little squares because we're not that same degree of magnification. And I've never seen this because they burst so fast. If you're doing this and if you have a real-time camera on the microscope, you might see them swell, but they burst very quickly. And that's everything for this presentation. I'm going to start and talk about active transport, which involves um, endocytosis and exocytosis and the sodium potassium pumps. And I think they're all worth just a quick way to remember them for the sake of the course. And then we'll look a little bit at the resting membrane potential, and that pretty much covers a lot of the next PowerPoint. And the one on all the organelles, I'll let you read. That's PowerPoint C. And PowerPoint D is the most important of this. And that's the one that involves the cell cycle and a little bit about transcription and translation. Thank you very much. And if you have any questions, they're easy enough. If I'm the instructor for this online course, my email will be available to you. Please contact me. So thanks again. I include these as YouTubes, by the way, that you will see when I incorporate them. I'm using the Google Meets presentation now, but I flip it to a YouTube and add it to our site. Thank you.